Hi, we are continuing with cell biology and life science in a nutshell. We were going over gluconeogenesis, which is the glyoxylate cycle, which is a variation of the tricarboxylic acid cycle. This occurs in plants, bacteria, protists, and fungi, not really in animals. And this is important for utilizing uh, carbon compounds, simple carbon compounds, as a source of carbon for making complex molecules when complex sources such as glucose are not available. This cycle generally occurs in germinating seeds of plants. It utilizes five enzymes associated also with the tricarboxylic acid cycle. These are citrate synthase, aconitase, succinate dehydrogenase, fumarase, and malate dehydrogenase. So, in the glyoxylate cycle, simple carbon compounds are used in the synthesis of macromolecules, including glucose. Fatty acids from lipids are commonly used as an energy source by vertebrates as fatty acids are degraded through beta oxidation into acetate molecules. This acetate bound to the active thiol group of coenzyme A enters the TCA or citric acid cycle where it is fully oxidized to carbon dioxide. So fatty acids from lipids lead to carbon dioxide. This cycle, the TCA or citric acid cycle, allows cells to obtain energy from fat. So utilizing this acetate from fat for biosynthesis of carbohydrates. And for the glyoxylate cycle, whose initial reactions are identical to the TCA cycle. Cell wall containing organisms like plants, fungi, and bacteria require large amounts of carbohydrates during growth for the biosynthesis of complex structural polysaccharides such as cellulose, glucans, and chitin. In these organisms, in the absence of available carbohydrates, for example, in certain microbial environments or during seed germination in plants, the glyoxylate cycle permits the synthesis of glucose from lipids via acetate generated in fatty acid beta oxidation. The glyoxylate cycle bypasses the steps in the citric cycle where carbon is lost in the form of carbon dioxide. The two initial steps of the glyoxylate cycle are identical to those in the citric acid cycle or TCA, acetate, citrate, isocitrate. Um, so in the next step, catalyzed by the first glyoxylate cycle enzyme isocitrate lyase, isocitrate undergoes cleavage into succinate and glyoxylate. The latter gives the cycle its name. Glyoxylate condenses with acetyl-CoA, a step catalyzed by malate synthase, yielding malate. Both malate and um, oxaloacetate can be converted into phosphoenol pyruvate, phosphoenol pyruvate, which is the product of phosphoenol pyruvate carboxykinase, the first enzyme in gluconeogenesis. The net result of the glyoxylate cycle is therefore the production of glucose from fatty acids. The net result, succinate generated in the first step, can either can enter into the citric acid cycle to eventually form oxaloacetate. So again, um, simple carbon compounds such as fatty acids from carbon from fatty acids are used for making glucose or complex macromolecules needed for plants, for fungi and bacteria. Acetate from fat is used for the biosynthesis of carbohydrates via the glyoxylate cycle. In this way, obtaining energy from fat via this pathway. So fatty acids are degraded through beta oxidation into acetate molecules, as lipids are used as an energy source by vertebrate. The biosynthesis of complex polysaccharides are result. So some of the organisms are shown which participate in this kind of cycle of making energy. So green algae, cyanobacteria, fungi, and plants. This is where gluconeogenesis occurs, a metabolic pathway resulting in generation of glucose from certain non-carbohydrates. This occurs only in non-animals, as shown in these pictures. So to finish off this section, this is an example of an animal, a newt lung cell stained with fluorescent dyes undergoing the early anaphase stage of mitosis. So an animal cell. Animals, again, are eukaryotic multicellular organisms. So they have a cell membrane. They form and a nucleus with the nuclear membrane for their genetic material. They form the biological kingdom of Animalia. Um, fatty acid beta oxidation affects long chain 3 hydroxyacyl coenzyme A hydrogenase deficiency. This is the process in a fat cell of lipolysis. So, showing a free fatty acid and how it is via ATP use changed to acetyl CoA and etc. So this is the diagram.
Fatty acid.
So this is the mitochondria. So the blood cytosol. So let's see. So this is where the glyoxylate process takes place. Sorry for the diagram. It's a little bit hard to see it. But the peroxisome and as well as the mitochondria are involved in this process of the gluconeogenesis. So as we mentioned, complex carbohydrates are formed as a result of this process of gluconeogenesis. Cellulose is an organic compound with the formula C6H10O5. It's a polysaccharide consisting of linear chains with triple strand of cellulose shown here. The hydrogen bonds are the blue lines between glucose strands. Cotton fibers represent the purest natural form of cellulose, containing 90% of the polysaccharide. Cellulose